Good morning students. This is lecture 34 under module 12. As you know, this is the last module and we have 3 lectures in this, and today's lecture is dedicated to facilitated transport. To understand what is the mechanism of facilitated transport, what is a carrier, what are the different types of carriers available, and what is active and passive transport, and a few applications. So, let us now understand what is actually facilitated transport means. So, it is a membrane transport process where there is a carrier agent which interacts specifically with a substance in the feed, substrate in the feed, and the substance carrier complex diffuses across the membrane. As you can see in this particular figure, this is a facilitated transport mechanism. So, this is also known as a carrier mediated transport, because there is a carrier inside the membrane which is actually binding to the substance or a substrate or the solute which we want to separate through the membrane, and then takes it across through the membrane and finally dispose it off at the permeate side. So, the carrier dissociates at the end of the membrane and finally returns to its original position. The exterior substrate concentration has no effect on the rate of transport. Usually, a carrier with high association and dissociation rate constants, which are similar in magnitude, is desirable. So, if I just try to draw something, let us say a small example, let us say this is the membrane, okay, this is the membrane, okay, and there is a solute X which I want to transport through the membrane using this carrier mediated transport, okay, and there is a carrier which is inside this membrane. Now, what will happen in this particular mechanism? This solute, okay, this solute will bind to the carrier. So, this is carrier, delta is carrier which is inside the membrane. So, the solute will come and sit here on the surface of the membrane, that is the interface of the membrane and the aqueous media, then it will bind this. So, once it binds, it becomes a complex, right, then it moves to the opposite side of the membrane or the permeate side. Once it comes here, by virtue of the concentration difference of the solute which you want to transport here, it will dissociate itself. So, X comes to the permeate side and that carrier becomes free. Now, again carrier is being recycled. So, this is how the facilitated transport or carrier mediated transport happens. So, we will understand the mechanism and the other aspects in detail. For the transport to be selective, the membrane is permeable to the flow of certain substances and impermeable to the flow of others, including carrier molecule. Now, carrier should be in such a way that its association and dissociation rate should be very high for the solutes, and it should remain inside the membrane, it should not come out out of the membrane. So, substances such as amino acids, organic acids, sodium hydroxide, sodium chloride, carbon dioxide, oxygen, metals and various ions such as cadmium, copper, cobalt and iron can be separated by using suitable carrier agents in liquid or solid composite membranes. So, this is a example of a facilitated transport in a liquid membrane. It is very easy to impregnate carrier molecules inside the liquid membranes, because in the liquid membranes are always so if you recall the liquid membrane structure. So, let us say this is pores, okay, this is another pore. So, the pore is already filled with the liquid membrane. So, you can impregnate your carriers here in a very easy way, it can be done. So, you can see how it is happening here. So, the carriers, this is the carrier which is impregnated inside the membrane, liquid membrane, this is the membrane, this is the membrane phase, this entirely. The solute that is going to be separated is coming to the liquid, and this is the liquid membrane and the bulk interface, you can say the feed interface, okay. Then it is crossing the membrane, okay, and it is getting bound to the carrier. So, you can see this is a carrier and this one here solute complex. Now, by virtue of the concentration difference here, so this carrier complex moves across the membrane. When it reaches here, this side, receiving phase side. So, your solute will be getting dissociated and your carrier again gets back, or it gets renewed basically, right. So, this is how this happens, the facilitated transport happens. So, 3 types of facilitated transport membranes are available. So, first one is mobile carrier membrane. Okay. So, here the carrier can diffuse freely through the membrane. You can see here how it actually looks like. So, this is the membrane actually, right. So, this is the liquid matrix in the case of the pores filled with liquids. This is your carrier and this is what is your target permeate, or you can say the solute, right. So, usually this type of membranes are liquid membranes. So, the major ingredient is often carrier solution or carrier bearing liquid compounds such as ionic liquids. So, ionic liquids are a class of green solvents which are very famous for their various excellent properties, or you can say that fine and tunable properties. We can prepare a particular ionic liquid to suit a target or for a particular application. So, the carrier here serves as a ferry boat plying between the two sides of the membrane. So, it is just moving here and there. You can see this carrier is moving here, okay. Again then it moves back to the other side after releasing the permeate. So, this is one. The second one is semi mobile carrier membrane. So, here the membrane is a polymeric membrane. You can see this is the polymer chains, these are polymer chains, this is your carrier and this is again your permeate, or you can say the solute. So, these are solid state polymeric membranes of which are often utilized as the hosting matrix, that mostly polymeric membranes. So, the mobility of carrier lies in between a mobile carrier and a fixed site carrier. The carrier can migrate elsewhere at a cost of high diffusional activation energy. So, the next one is fixed site carrier membrane. So, here also again the membrane is a polymer matrix. You can see the polymer chains here, these are your carrier, this is the solute or permeate. So, usually solid state membranes, carrier can only vibrate within a confined nanospace rather than migrate it, because you can see here the carrier is fixed to the polymeric matrix. You can see it is fixed, okay. You can see something like this, the structure. So, this is fixing structure, right. So, the target molecular ion passes through the membrane by carrier to carrier hopping. So, this mechanism is called hopping mechanism. We can let us say how it will happen. So, let us say this, it comes here, okay, the solute. Now, it comes here and binds here, right. Once it binded, after some time and then it hops again here, then it hops again here, then it comes and get dissociated in the opposite side. So, basically it is a hopping mechanism that takes place in a fixed site carrier membrane. So, let us understand the mechanism, how the facilitated transport actually happens. So, let us take an example of a species A which moves through the membrane, okay, and C is the carrier species. So, A is the solute which you want to remove or transport using the membrane, and C is the carrier. Now, what is happening? The C is reversibly react or bind with A in the feed site of the membrane to form a permeate carrier complex. So, this is the membrane, okay, and this is the membrane feed interface. So, here this reaction is happening. So, A plus C is giving Ca. So, Ca is your it is a intermediate complex, right. So, then this moves across the membrane in this direction because of the concentration gradient, you can say. Once it reaches the permeate side, okay, now it releases and this reaction happens. So, Ca it will give you A and C. So, A is retained here and C is recycled back to the membrane. So, the complex Ca diffuses through the membrane, reaches the permeate side where it decomposes and releases the permeate A. So, the carrier C gets freed and diffuses back to the feed site where it again binds to a fresh permeate A. So, C acts as a shuttle within the membrane. So, it is shuttling, only C is free inside the membrane matrix. So, it is shuttling between the feed side and the permeate side. So, the component Ca decreases across the membrane and that of the carrier, and that of the carrier has a concentration gradient in the opposite direction. You can see that Ca is this is the gradient here, this is Ca, okay. It is decreasing this one with respect to across the thickness of the membrane, you can say, okay, whereas the carrier is in the concentration profile or gradient is in the opposite direction. So, both Ca and C diffuses across the membrane under their respective concentration driving force. So, the polymer matrix can also contain a functional group that act as a carrier called as a fixed carrier, right. We have just discussed about a fixed carrier membrane. So, for this case the solute transport occurs by hopping mechanism, very similar to that of a surface diffusion. Facilitated transport is much more faster than passive transport because of the higher effective solubility of the Ca complex in the membrane. So, if the fluid in the permeate site contains another species B, So, we just discussed previously about one component, the feed contains only one solute, that is let us say which we discussed as solute A. It is binding to C, the carrier, and it is getting associated, then the complex passes through the membrane matrix, then dissociated itself in the permeate side. Let us now understand that if there is a component B, another solute which is present in the permeate side, okay, then what will happen? That relieves A from the complex C A, the flux C A further increases by this phenomena. That means, what is happening? The complex B A does not diffuse through the membrane. Okay, though B and A is forming a complex, it is not diffusing back to the membrane. So, you can see the reaction feed side is basically A plus C is giving AC, then this complex is moving through the membrane, then once it reaches the permeate side. So, this complex AC plus is reacting AC plus B is giving BA plus C. So, but BA is not diffusing back, but the C is again recharged or renewed. So, it is free now to bind with another solute of A. So, the species A is recovered by decomposing the complex BA leaving from the permeate unit, B is again recycled to the permeate side fluid. So, the concentration of A in the form of BA in the permeate side may be higher than that of the feed side. For such cases, facilitated transport of A occurs against its apparent concentration gradient. However, we do not have to provide any extra energy for this to happen. So, let us understand the three important characteristics of facilitated transport are. The first important thing is that solute flux. So, solute flux should be larger and it should be highly selective or more selective. Second is that at what happens at low solute concentration, usually the flux varies linearly with solute concentration. However, once it reaches a very high solute concentration and it gets saturated after reaching a limiting value. So, the third one is that flux of one solute may easily poisoned by another solute or may be strongly influenced by the gradient of another solute. So, these are the three things that need to be considered while we design some facilitated membrane system. So, let us understand few applications, some real life applications like this facilitated transport of oxygen through the lungs of the membrane. You know the hemoglobin, the carrier is the hemoglobin here, it binds to the permeate oxygen to form oxyhemoglobin complex. Now, this oxyhemoglobin complex it diffuses through the membrane and once it reaches the permeate side, it will release the oxygen to the opposite side and again it is free now to bind to another molecule of oxygen. So, the presence of hemoglobin significantly increases the solubility of oxygen and boost its rate of transport through the membrane. So, increasing the solubility of oxygen in the presence of oxygen is something very important. So, this is how it happens actually, the oxygen is getting transported inside the cells. Another example is, it is a chemical industry example, separation of olefin from a gas mixture by silver as carrier, using silver as carrier. So, silver reversibly binds to olefin. So, you can see the reaction. So, silver plus olefin gives a silver olefin complex. Now, the silver olefin complex diffuse to the permeate side which is present in the lower pressure and decomposes there to release the olefin. So, in the permeate side what is happening, the complex is again dissociates to silver and olefin. Now, the regenerated silver diffuses back to the feed side through the pore membrane filled with a solution of silver salt. So, this is the schematic illustration of how the pi complexation bond formed between olefin and silver. So, you can see how the reaction is happening. It is not a reaction, basically you can say the bonding between silver and olefin. Another example is separation of carbon dioxide from air by amine functional group carrier. Now, carbon dioxide capture has become a global concern due to the anthropogenically forced carbon emission and climate change. 2 mole of polyethyleneamine absorb 1 mole of carbon dioxide. So, this is the membrane basically or membrane material you can say. So, by using PEI, numerous NH functional groups which are already present in the membrane matrix reacts with carbon dioxide to form carbamate or bicarbonate under humid conditions. So, a humid condition is the necessary requirement. So, carbamate will be hydrolyzed by water to regenerate amine on the opposite side, So, you can see how it is happening. So, this is the schematic of carbon dioxide and nitrogen transport mechanism in a PI facilitator transport membrane. So, you can see the dashed lines, these lines. So, it is actually the pathway for nitrogen gases. So, the nitrogen is getting transported whatever amount, okay, which is much lesser than that of the carbon dioxide, of course, and the mechanism for this transport is solution diffusion, whereas carbon dioxide is getting transported through the facilitated transport by combining or by binding with the NH groups already present in the membrane. So, when it reaches here, the carbon dioxide, okay, will dissociate from the NH groups and the amine will be regenerated. So, these are the various reactions, you can just go through later on and see understand what are the different reactions that is going through when carbon dioxide is getting transported through the membrane using a facilitated mechanism, right. So, now let us understand what is coupled transport. So, coupled transport resembles facilitated transport, except that a complex BC forms on the downstream surface of the membrane by reaction with a species present on the permeate side liquid. Now, a kind of active transport across a biological membrane in which the transporter protein couples the movement of an ion, usually or you can say typically sodium or hydrogen, down its electrochemical gradient to uphill the movement of another molecule or ion against a concentration or electrochemical gradient. So, energy stored in the electrochemical gradient of an ion is used to drive the transport of another solute against a concentration or electrochemical gradient. The free energy needed to perform active transport is provided by the concentration gradient of the driving ion. So, sodium is the driving ion for many co-transporters and exchangers. These transport proteins may also be referred to as sodium coupled co-transporters. So, this is an classic example of a coupled transport or secondary active transport. Here you can see that there is a this x and y's, okay, x and y here. So, x and y represent transporter substrates, okay, that is getting transported. So, a example is sodium plus glucose co-transporter found in small intestine and kidney proximal tubules simultaneously transports 2 sodium ions and 1 glucose molecule into the cell across the plasma membrane. So, this is how it happens. You can see here how it is happening by different mechanism. One is called antipod, another is called sympod. This antipod and sympod we will discuss at the fag end of our today's lecture. So, here you can see that sodium is binding, okay, and then getting transported inside the cytoplasm, and here sodium is again binding to the X and getting transported to the cytoplasm, and here sodium and potassium, sodium is coming out of the cytoplasm, whereas potassium is getting inside the cytoplasm by supply of certain energy in the form of certain energy. So, it can be ATP or it is ADP, right. So, another example is Na plus and Ca2 plus exchanger, cation and calcium exchangers. So, found in cardiac muscle cells and elsewhere in the body, transport 3 sodium ions into the cell in exchange for 1 calcium ion transported out of the cell. So, that means what is happening in this in the exchanger mechanism of sodium and calcium. So, sodium is getting inside the muscle cells, cardiac muscle cells, whereas calcium is getting out of the muscle cells. So, let us now understand the coupled transport mechanism. So, in coupled transport, a species B present in the downstream side of the membrane form a complex BC by reacting with the complex Ca present in the permeate side. So, you can see that A plus Bc, this is the reaction, A plus Bc is giving you B plus Ca. So, the Ca is getting transported across this membrane in this side, across the length of the membrane. Then once it comes to the permeate side, Ca is binding to B, which is already present in the permeate side, thereby releasing A and forming another complex Bc. Now, this Bc is moving in the opposite direction. So, this is called coupled mechanism. So, Bc diffuses through the membrane and reaches the upstream surface, where it again reacts to the surface species A by forming a complex Ca and releasing B. This is how it happens. So, what is happening here? So, the transport of A is happening here, okay. Transport of B is happening this side, okay. So, in the by this process the complex Ca is formed on the upstream side that diffuses through the membrane to the permeate side. So, A plus B Bc giving Ac plus B. The species Ca and Bc diffuses through the membrane in opposite direction. In overall, the species A travels from the feed side to the permeate side and species B travels from the permeate side to the feed side, because of which the phenomenon is called the coupled transport. So, it is a coupling effect basically. So, the concentration of Ca is the gradient is in this side, okay, and concentration of Bc is gradient is in this side. So, that is why it is moving, Bc is moving in this side, this direction, and Ca is moving in this direction. So, now a few applications. So, the first one is copper recovery from waste liquor using a microporous membrane. So, the membrane used in this particular application is a microporous polypropylene of waste cleanest membrane called Celgar, impregnated with a solution of Leak 64. So, Celgar is the membrane, okay, Leak 64 is the carrier. So, Leak 64 contains an enzyme dissolved in an organic medium immiscible with water. So, enzyme is the carrier. So, in the feed side where the pH is maintained as 2.5, the enzyme, that is the carrier, forms an complex Cu copper enzyme complex by binding with copper. So, the copper 2 plus and enzyme H plus is binding to, is giving you copper enzyme complex plus hydrogen. So, the complex diffuses to the permeate side where the permeate side pH is maintained as 1 through the liquid filled pores of the membrane. The complex reacts with hydrogen ion on the permeate side and form protonated enzyme by releasing the copper. So, the copper enzyme complex plus proton or hydrogen, which will give you the copper as well as enzyme hydrogen complex, which will again diffuse back. The protonated enzyme diffuses back to the feed side where it releases the hydrogen ion at the higher pH. The free enzyme again bind with another copper and the process is repeated and it's continued. Now, a carrier agent. So, carrier, since this is a facilitated or carrier mediated transport. So, carrier, what is a carrier, how to choose a carrier, what are the properties of carrier, this plays a very important role. So, in facilitated transport in membranes, a carrier agent interacts specifically with a substrate in the feed mixture and form a substance carrier complex. Now, this complex diffuses across the membrane and the carrier dissociates at the end of the membrane and finally return to its original position. So, just we have discussed again oxygen transport through the cells, oxygen in the presence of hemoglobin. So, oxygen plus hemoglobin is forming a oxyhemoglobin complex. This complex diffuses through the membrane, this is the complex, okay, right, and then it releases oxygen here and the hemoglobin is back to business, right. So, the carriers are basically classified based on several specific target permeates, as for example, whether it is carrying oxygen, it is a oxygen permeate carrier, or the carbon dioxide transporter or carrier, olefin, sugar, etc. 4 basic types of reversible reactions happens. So, those are proton transport reaction, nucleophilic addition reaction, pi complexation reaction and electrochemical reaction. Now, carriers and the corresponding target permeates are therefore closely connected with reversible reactions and the ligament. So, these are 4 types of 4 typical types of reversible reactions which is listed here, right, for facilitated transport membranes together with their corresponding carriers and target permeates. So, the in a proton transport reaction, if Bronsted base is the carriers, then target permeates can be hydrogen, hydrogen sulfide, ammonia, etc. In a nucleophilic addition reaction, if Bronsted acid is the carrier, then carbon dioxide, sulfur dioxide, COS, etc. can be permeates or can be transported. So, if in case of a pi complexation reaction, metal ion or charged metal atoms are basically the carriers. So, olefins, arenes, thiophene, etc. are the permeates. So, in case of electrochemical reaction, so this oxygen 2 minus or electron, they are the carriers and usually the oxygen is the permeate. So, let us now understand what is active transport. Now, active transport, also known as uphill transport, is a process of transporting molecules from a low concentration region to a high concentration region. So, active transport must couple to another spontaneous process to transport substrates against their thermodynamic forces. It is a very important process for most of the biological systems. Various artificial membranes are used for the active transport of substances. Since the process is not spontaneous and sufficient amount of input energy is required, so that active transport can happen. Now, there are few examples I have just listed down. So, the first one is transport of sodium and potassium ions to the cell membrane of the red blood cells against their respective concentration gradients. The second is transport of glucose from blood. So, usually it is in the lower concentration as blood to the liver cells, where glucose is already presented in higher concentration. So, it is against the concentration gradient. Then transport of minerals in the soil, which is present in the lower concentration to that of the root tissues of the plants, where the minerals are present already in higher concentration against the concentration gradient. So, the mechanism of such transports are very complex. The mechanism may be resembled to the pumping of a liquid by squeezing the silicon tubing of a peristaltic pump by its roller. So, how it happens now in peristaltic pump? Let us say this is a peristaltic pump. Okay. The roller. So, the silicon tubing is coming like this, right, and then again there is another roller and again. So, you are pumping here some material. So, it will come here and again it will, the two rollers will be here. Okay. So, it will squeeze, here it is getting squeezed. So, you can see some structure something like this. Okay. So, what will happen in this? So, this is the schematic. Okay. Whatever the flow is getting, that is squeezed, and it is getting squeezed, then it will be expanded. So, basically again it is a throwing mechanism, squeezing out, squeezing in, squeezing it, squeezing in, squeezing out, something like that. Okay. So, energy required for such transport is supplied by ATP, which is adenosine triphosphate, maybe ADP also. So, now let us understand how the cations transport happens. As I told you earlier about symport and antiport, here we will try to understand actually what is symport and antiport. So, two types of mechanism for cation transport. One is symport, which is also called a co-transport, another is called antiport, which is called exchange. There is something called uniport. So, uniport in the case in which one permeate is actually getting transported in a single direction, okay, inside a membrane. But we will try to understand what is symport and antiport. Now, you can see the basic differentiation in the diagram itself. In symport, what is happening? The transport proteins responsible for coupled transport are referred to as co-transporters, also known as symporters, okay, and exchangers, also known as antiporters. Okay. So, in symport, the direction of transport is the same for both the driving ion and driven molecular ion. Okay. So, here both are both A and B are driving in the same direction, but in antiport it is the reverse. That is why the name antiport actually. The driving ion and driven ion on molecule are transported in opposite direction. So, we will try to understand actually how symport happens. So, I will try to draw a schematic. Okay. So, let us say this is here membrane, okay, right, and this is a phase, aqueous phase 1. Phase 1, here I have a phase 2, we can say aqueous phase, of course, it is aqueous phase, right, and we will talk about how the cation transport is happening in a symport. Okay. So, initially what will happen? So, salt, okay, plus X minus, okay, it will bind, it will come to the interface here, okay. The phase 1, the interface of the membrane and aqueous phase 1, and it will complex here, right. So, once it is complexed here, complexed with the carrier basically, here we will talk about a carrier which is usually neutral. So, I is a neutral carrier, okay, right. So, what will happen? So, this I is coming here and it is binding to what? Binding to M, okay. So, now it is forming a complex which is called I M plus X minus complex. Now, once it reaches here, right, it reaches here, okay, to the opposite side of the membrane. So, the phase 2. So, the phase 2 interface, it will release the salt M plus X minus and the I is getting renewed. Okay. So, your M plus X minus both are getting transported in the same direction, whereas I is bounding only to the cation. Okay. So, this is how it happens in case of here Na plus glucose system. Okay. So, your sodium is bound to one of the carrier protein, and along with that glucose is getting transported. Okay. So, it is very this one common in most of the cells, human cells, animal cells. Okay. So, once the carrier leaves the complex, releases the salt that side. So, it gets renewed and comes back to the membrane side, inside the membrane, and again get associate itself with another molecule of the salt, form a complex, and again the process continues. So, this is in case of sympod. So, in case of antipod, okay, let me try to explain again by drawing another image. So, this is your membrane. Okay. So, this is aqueous phase 1, this is aqueous phase 2. So, here we can have a negatively charged this one here carrier, let us say that is negatively charged carrier I minus, okay, and there is a salt which this one a cation which I want to transport, as there is a another cation also. Okay. We are talking about antipod. So, in the opposite side. Okay. So, what will happen, this M, okay, it will complex with the I and form a complex I minus M plus, then it moves across the membrane due to the concentration gradient. Once it reaches the aqueous phase interface 2 here, it will release your M plus, right, and it will bind to this N, thereby it will form another complex called I minus N plus complex. Now, this I minus M plus complex will reach here and it will release here N plus. Okay. So, I hope you will understand how it is happening. So, here M plus is moving in this direction, okay, right, whereas here N plus is moving in this direction. Okay. And please try to remember that they are binding to one particular negatively charged carrier molecule, okay, one single type of carrier, that is binding to first the cation that is present in the aqueous phase 1, releases it in the opposite side, again bind to the cation which is present in the opposite side or the permeate side, from the complex brings it back to the upstream side or feed side or aqueous phase 1 side, dissociates, again binds to the cation present here, this M plus. Okay. So, here actually it is you can note that so two molecules are transported in opposite direction. So, and you are using one anionic charged or negatively charged carrier or anionic carrier. So, that is very important if we are talking about transport of cations. So, the single cation itself is a single anionic carrier is binding to both the cations, and this is how your antipod reactions or transport happens. Now, these are the examples of active transport systems. So, you can see the permeate, carrier and membrane all are given here. So, if you want to talk about uranyl ions, so we will have a carrier, this is a big name actually 2,3 epithiopropyl methyl cryolate dodecyl. Okay. It is a big name actually. So, and we can have an anion exchange membrane. So, in case of transport of sodium and potassium, so oxidative phosphorylation will be the carrier, living cell mitochondria is the membrane, and in case of anions, reversed bi-enzyme is the carrier, porous membrane can be the membrane. So, similarly there are other examples also, you can go through it later on, right. So, now understand the active transport applications. So, sodium potassium pump. Now, what is this? This is for real life in real life cells. So, exchanges what it does actually, it exchanges sodium ions for potassium ions across the plasma membrane of animal cells, okay, right. So, how the mechanism happens actually, it is very clear in the this particular image. So, sodium is getting transported in this side, right, whereas here potassium is getting transported in this side. Okay. So, this is how it is getting transported. 3 sodium ions bind with the protein pump inside the cell, right. Then what happens, the carrier protein gets energy from ATP and it changes its shape. So, in doing so, it pumps the sodium ions out of the cell, right. So, here these are the pumps, you can call these are the pumps, right. So, at that point 2 potassium ions from outside the cell bind to the protein pump, then potassium ions are then transported into the cell and the process repeats. So, you can remember that again this is something like that, this antipode mechanism which we have just discussed, that 3 sodiums are getting exchanged with 2 potassium ions, right. So, the energy that is required for doing this transport is comes usually from the ATPs, okay, the adenosine triphosphate, or ADPs, adenosine diphosphate, and the PIs. So, the another example is the sodium glucose transport protein. This we have discussed little earlier, but just for another time. So, this is a secondary active transport system. The protein binds to 2 sodium ions which want to move into the cell and 1 glucose molecule which wants to stay outside of the cell. So, the diffusion of the sodium into the cell allows the glucose to also be carried out into the cell without the transport protein expending ATP. So, ATP must be expanded by the sodium potassium pump elsewhere in the cell to keep up the sodium gradient that allows the sodium glucose transport protein to function. There should be a gradient of sodium, otherwise sodium would not at all move. So, this is how the sodium and glucose transport happens. So, another example which is very classic is of course, the mineral transport from soil to inside root of a plant. So, what is happening here, you can see this particular figure actually. So, during the active absorption of minerals, so ions from the outer space of the cell move into the inner space and it generally occurs against the concentration gradient. Now, these move into the roots by using energy in the form of ATP. Specific proteins present in the membrane of the root hair cells actively pump ions from the cell to the cytoplasm of the epidermal cells. Now, these proteins act as a control points for the type and quantity of the solutes, quantity of the solutes that reach the xylem. So, you can see the mineral ions enter by active transport. Okay, water enters the roots and root hair by osmosis. Water and ions pass up through xylem and then transpiration and all these things happens. So, now let us understand what is passive transport. Now, we have understood what is active transport. Now, let us discuss what is passive transport. In passive transport, compounds are able to move freely across the membrane based only on a favorable concentration gradient. Requires no energy for transport of compounds. So, basically 3 types: simple diffusion, facilitated diffusion, and osmosis. You can see how it is happening here. So, this is simple diffusion of lipid soluble molecules through plasma membranes. So, oxygen is moving in this direction and carbon dioxide is also moving in this direction, okay, water also moving in this direction. So, this is actually in both side it is happening. So, this is your simple diffusion mechanism. So, in simple diffusion the molecules move from an area of high concentration to area of low concentration. So, that is all, that is how it is only happening using the concentration gradient. So, molecules naturally flow down their concentration gradient, diffusion proceeds without an input of energy. Molecules that are small and uncharged can freely diffuse across the cell membrane. So, one example is movement of oxygen from lung cells to blood due to concentration gradient. So, the next is facilitated diffusion. Here it involves the passive movement of molecules along their concentration gradient guided by the presence of another molecule, usually an integral membrane protein forming a pore channel. Okay, this is how you can see the pore channel is formation happens, right, and how it binds to the carrier proteins and then getting transported. So, it does not directly involve high energy molecules like adenosine triphosphate or guanosine triphosphate GTP, since the molecules are moving along their own concentration gradient. So, example is sodium ion crosses the membrane through a channel protein specific for. The next is osmosis. You know what is osmosis, we have discussed osmosis reverse osmosis in great details. So, diffusion of water through a semi-permeable membrane according to the concentration gradient of water across the membrane. Only water is transported across the membrane and water limits the diffusion of solids in the water. If water has a concentration gradient in this system, it will diffuse down its concentration gradient, crossing the membrane to the side where it is less concentrated. So, osmosis continue until the concentration gradient of water goes to 0. This is how your osmosis is happening. So, I think we will just discuss one example, osmosis example. So, both animal cells and plant cells lose and gain water by osmosis. Okay. This is how osmosis is very important phenomena for all the living things. So, inside ear cells and outside ear cells, what is happening, you can say it is a semi permeable cell membrane. This is the membrane, okay, this one, and water, this is water, this is the big one is salt. So, movement of water molecules is happening from outside of the cell to inside of the cell, okay, and along with that of the salt. So, water moves from an area of high concentration, that is outside the cell, through the cell membrane to an area of low concentration inside the cell. So, plants rely on osmosis to take in water through their root hair cells. These cells have a large surface area that efficiently absorb water and other different types of minerals. So, when a plant is watered, the water is absorbed, moves from an area of high concentration, that is soil, to an area of low concentration, that is the root hair cells, before being transported to the rest of the plant. So, this is how osmosis is taking place in root hair cell. So, I think with this we will conclude this today's lecture. Most of it is being taken from Professor Nath's book and B.K. Datta's book and some other this one reports or this one papers from literature. So, you can refer them later. So, with this I thank you very much. In case you have any query, please feel free to write to me at kmohanty@iitg.ac.in. Our next lecture, that is lecture 35 under module 12, will focus on different types of gas liquid and liquid contact devices. Then we will discuss little about membrane reactor and bioreactors and PEM hydrogen fuel cell. So, thank you very much.